This one looks terrifying, right? But let's go through it carefully. So the heading on this overhead and the previous one is NMDA receptors are coincidence detectors, okay? I'll explain what this means. So first of all, on the first page, it showed a low level of stimulation, right? Like something is exciting the presynaptic, neuron and provoking it to release a certain amount of glutamate, right? And there's activity at these, ionotropic receptors, and there's excitation and maybe action potentials in the next cell. But let's say something really dramatic happens, like a lot of activity is occurring along that presynaptic, cell, and a lot of glutamate is being released into the synaptic cleft. Think of it like if you have a complex network where you have, one axon terminal coming in and then another axon terminal coming in, and then you've got your postsynaptic cell, right? This is your postsynaptic membrane. If only one of them is firing, right, you'll get a certain level of activity. But if both of them are firing, you'll get a lot more glutamate out there, right? And as you get a lot more glutamate out there, you're going to get a lot more of these ionotropic receptors activated, right? They're going to open. You're going to get a lot more permeability of the membrane, a lot more holes to allow for sodium to pass through. And as sodium comes in, you're going to get a lot more depolarization, right? It's going to go more depolarized than it was previously under low levels of stimulation. And when there's a lot of depolarization in here, this magnesium ion is actually, it's a positively charged ion, right? It actually gets ejected from the channel. So at high levels of stimulation, this NMDA receptor will open and allow for the passage of sodium and another ion, calcium. So at high levels of depolarization, the magnesium ion is actually ejected and the NMDA channel is now open, okay? And calcium, it turns out, is a pretty potent messenger inside the cell. It can activate, much as G proteins sort of activate, all sorts of second messengers and sort of stimulate activity on all of these biochemical cascades that can reach the nucleus, right, of the cell and the DNA and produce changes in which genes, in terms of which genes are expressed and therefore which proteins are generated. So second messengers get activated by the entry of calcium, and this changes the physical structure, right, of these synapses. It turns out that along glutamate synapses, right, it tends, high levels of activity tends to strengthen connections, sort of upregulate the postsynaptic response, the response of the next cell. It's kind of like the idea of use it or lose it. If there's a lot of activity occurring in a synapse, a glutamatergic synapse, you're going to actually get these NMDA channels open, you're going to get calcium to come in, you're going to get changes in gene expression, and you're going to strengthen that connection. You may even grow additional connections at this location to really make the postsynaptic membrane more responsive to presynaptic stimulation by glutamate.